Okay, our next speaker is Yizhou Dong. He's a professor at the Icahn School of Medicine at Mount Sinai, and his research focuses on the design and development of biotechnology platforms for the treatment of genetic disorders, infectious diseases, and cancers. How's it going there? Are you able to get your screen up?
Great. So good afternoon, everyone. Thanks so much for the kind introduction, and also thanks so much for having me. Let me share my screen. Okay, so it's really my great pleasure to share with you some of our recent studies on lipid nanoparticles and apply that to very diverse therapeutic applications. So first, I would like to give you an overview about research in my lab. We are focusing on the development of platform technologies, which can be applied to diverse therapeutic indications. And the first platform is to develop novel lipid or lipid-derived nanoparticles. We synthesize all kinds of functionalized lipids, and then we formulate them with different therapeutic cargoes, such as messenger RNA, siRNA, and other therapeutic components. And the second platform is to engineer RNA itself. We can engineer messenger RNA to improve the production of the functional protein, or we can engineer CRISPR system to help enhance the genome editing capabilities. And the next direction we work on is adoptive cell therapy. We can engineer different cell types and take advantage of their endogenous functions and help tackle unmet medical needs. So today I would like to discuss with you one example about our development of lipid-derived nanoparticles, and also one example engineer macrophages for treating bacterial infections. As we all know now, messenger RNA has become a very important class of therapeutic agents. With the very fast and quick success of the COVID-19 mRNA vaccines, we are able to control the COVID-19 pandemic within a relatively short time. And this vaccine platform can help induce the antiviral immunity and protect a broader public. And in addition to viral vaccines, mRNA has been applied to very diverse therapeutic indications in preclinical studies and also in clinical trials. So for example, mRNA has been applied to cancer immunotherapies, protein replacement therapies, genome engineering, and genetic reprogramming. So although mRNA has become a very important class of therapeutic agents, delivery is still one of the key challenges for its broad applications in human patients. Because mRNA is not stable. If we inject it directly into human body, mRNA would be degraded very fast within a short period of time. Therefore, we need to develop delivery particles to load the messenger RNA. Even with the protection of different type of carriers, such as lipid nanoparticles or polymers, there are still many biological barriers. So for example, shown here, if we inject the nanoparticles intravenously, these particles need to diffuse through the extracellular matrix, and also meanwhile avoid the filtration by kidney, uptaken by phagocytes, and then they are uptaken by the desired cell populations. And in the cells, mRNA need to be released to the cell cytosol. Then they can access the protein production machinery and then produce the functional proteins. So there are so many layers of biological barriers, and we have to overcome. So to address this delivery issue, we have to develop novel biomaterials. So based on our previous experience on the delivery of different type of RNA, we designed this particular chemical structure. It has a long chemical name shown here. and we just give it a short name, TT lipids. And these TT lipids are composed of a phenyl ring, 3 amide linkers, and multiple lipid chains. And we can tune the carbon length, we can tune the functional groups. Through this very simple one-step reaction, reductive amination, we can synthesize these TT lipids in large scale. This slide shows our overall strategy to develop and optimize these lipid-derived nanoparticles. We have the newly synthesized TT lipids, and we have several other formulation components. The helper lipid, such as phospholipid, cholesterol, these are natural components of the cell membrane. And also another lipid, PAC lipid, to stabilize the particles. And here the therapeutic cargo is messenger RNA. Through this very nice microfluidic-based device, these particles can form very nicely. And then we can study the particle properties, such as particle size, particle surface charge, loading efficiency of messenger RNA, and also protein production efficiency. And then we utilize orthogonal experiment design, which can help us to identify the optimal formulation with a significantly reduced experiment load. And this approach is widely used in the field of chemical engineering. We borrow this method in our optimization of the formulation ratios. And due to the time limit, I'll just skip the detailed process. And after we identify the optimal formulation, then we can test the formulation in different disease models. And we chose a disease model here shown hemophilia, which is a genetic disorder. It is caused by missing or mutation of factor 8 or factor 9. Patients suffer from life-threatening bleeding or serious complications such as joint bleeds. So in this study, we formulated the lead material TT3 with mRNA encoding human factor 9. Then we performed the intravenous injection first in wild-type mice. So on the left figure, the y-axis is human factor 9 concentration in mouse plasma. X-axis different treatment groups, including untreated mice, free mRNA-treated mice, irrelevant mRNA-treated mice, and also TT3 loaded with human factor 9 mRNA at 2 different doses. And you can see when we increase the dose, there is increased production of the human factor 9. Then we performed a similar study in Hemophilia-B mouse model, that is factor 9 knockout mice on the right. And you can see the results are consistent. You see still dose-dependent production of the factor 9 protein in mouse plasma. Next, we are wondering if these factor 9 proteins are active or not, and then we perform the activity assay. And here the y-axis, that's a human factor 9 activity in MIU per ml, and the x-axis different treatment groups, control, negative control TT3 with Firefly luciferase mRNA, and our treatment groups TT3 loaded with human factor 9 mRNA with 2 different doses. And at the 1.1 milligram per kilogram dose, the factor 9 activity is around 800 MIU per ml. In normal plasma, our human factor 9 activity is between 500 MIU to 1500 MIU. So that means that TT3 at 1.1 milligram per kilogram dose is able to fully recover the factor 9 level to the normal range. So the results are very promising, and currently these materials have been licensed by biotech companies for further development as a clinical trial candidate for treating diverse diseases. Next, I would like to switch our research topic on the adoptive cell therapy. So we are very interested in address the drug bacterial infections, especially drug-resistant bacterials. We know macrophages are one of the most important cell types in human body to fight against bacterial infections. But if for some drug-resistant bacterial, these macrophages cannot clear them just by their endogenous functions. So we are wondering if we can utilize certain approach to engineer macrophages to enhance their antimicrobial activities. Then we think about biomaterials. And when we walk in the pharmacy store, we always see a lot of vitamins. And as shown here, here is vitamin C. And the vitamins have very diverse functions. Biologically, they possess unique functions for different pathways. And chemically, they possess unique chemical functional groups, such as hydroxyl groups, hydrophilic groups, charges. All these are very interesting chemical functional groups. We think maybe we can modify those vitamins and make vitamin-derived lipid nanoparticles, and then engineer macrophages for treating bacterial infections. Then we found this disease, sepsis. And sepsis is a life-threatening condition caused by viral or bacterial infections. And each year, over 30 million people worldwide are affected by sepsis. Although with advances of the intensive care and antibiotics, sepsis is still the #1 cause of death in hospitals. Its mortality rate is very high, around 25 to 30%. And also each year, the death rate is increasing. This slide shows the challenges for treating sepsis, especially the late stage sepsis. About 70 to 80% of septic deaths is related to persistent infections. And more importantly, there are many drug-resistant bacterials, which make the treatment even more challenging. And for those septic patients, researchers found that their immune systems are significantly paralyzed, especially their macrophages, monocytes numbers are much lower than normal range. And for the past decade, there's no any new effective treatments have been approved by FDA. So we are wondering if we can design some new strategies to address this deadly disease for bacterial infected sepsis. Here's our overall strategy. We can design vitamin-derived lipid nanoparticles and deliver antimicrobial components into macrophages. And then we transfer these macrophages to here shown septic mice. And then these engineered macrophages can help eliminate drug-resistant bacteria and eventually recover the body conditions for these disease mice. To test our concept, we first need to design the vitamin-derived materials. And here are some examples. We synthesized vitamin-derived lipids here, such as vitamin B3, VC, VD, VH. We installed amino groups which can interact with messenger RNA under acidic conditions. And then we performed multiple rounds of the optimization and examination in macrophage cell models. Then we identified a lead material that's a vitamin C derived lipid nanoparticle formulation C5. And you can see it would over 70-fold better than vitamin 3000, over 300-fold better than electroporation. These are two of the state-of-the-art technologies for delivering RNA into macrophages. And on the right, we can visualize the particle morphology through electronic microscope. And you can see here the particle in the spherical morphology, and their particle size are relatively homogeneous. Now we have a pretty good delivery material for macrophages. Then we need to think about a therapeutic cargo. In this case, that's an antimicrobial component for treating bacterial infections. Then we go back to the literature. We try to learn how macrophages fight against bacterial infections. And we found when macrophages eat those bacterials, they first form an organelle called phagosome. And then those phagosomes are fused with lysosome to form phag lysosome. Normally, for some drug-sensitive bacteria, those would be cleared by the either antibiotics or the macrophages themselves. But for some drug-resistant bacteria, they figure out some approach to secrete proteins or toxins, and they can escape from those organelle and then further proliferate and infect other cells. So we are thinking maybe we can enhance the antimicrobial activity in macrophage lysosome, and then we can block the immune invasion of those drug-resistant bacteria and eventually eliminate those bacteria. To achieve that, we designed this particular messenger RNA, which has 3 components. One is antimicrobial peptide, AMP, and one component enzyme-sensitive linker, and one component cathepsin B, cat B. So these 3 components have their different functions. Cat B is an endogenous protein in macrophage lysosome. So after translation, this protein can translocate the whole protein into macrophage lysosome. And in macrophage lysosome, the enzyme-sensitive linker would be cleaved and then release the antimicrobial peptide. And this peptide is has a very strong antimicrobial activity that can work together with macrophage to eliminate drug-resistant bacteria. Then we produced this messenger RNA and tested in different cell models, bacterial infected cell models, and we found that they did show very nice antimicrobial activity. And then we moved to animal models to test the efficacy. We established a sepsis mouse model. This is well established in the literature, and we try to mimic the human sepsis condition. Therefore, we used multi-drug resistant bacterial infections, and also we used mixed bacterial S. aureus and E. coli. And on the left, the y-axis, that's bacterial load, and the x-axis different treatment groups. And we can see with the engineered macrophages that can significantly reduce the bacterial load compared to the other 2 control groups. And more importantly, this engineered macrophages can greatly improve the mouse survival. Here you can see over 80% mouse survival compared with the other 2 control groups. Then we quantified the mouse conditions. We found all the survived mice, their body weight continued to grow. And we also measured white blood cells and lymphocytes. And you can see after 30 days, both their white blood cells and lymphocytes return to the normal range. So in conclusion, we briefly talk about our process for the development of lipid nanoparticles and for delivery of different type of mRNA. And we showed a proof of concept study for treating Hemophilia-a. And in the second part, we talked about adoptive macrophage therapy, and with the vitamin C derived lipid nanoparticles to engineer macrophages. This cell therapy can help improve the survival rate of the septic mice and recover the body conditions of those septic mice. So in the next a few years, our lab together with our collaborators, we work on different fields of materials, on engineering, gene editing, immunoengineering, and cell therapy. Our goal is to translate our scientific findings and technology development into more effective treatments for patients in the clinic. So lastly, I would like to acknowledge a lot of people. We have a talented group of lab members, including postdocs, graduate students, undergrad students, and several high school students. Also, thanks our collaborators for their significant contributions for different projects. Without their help, we cannot make a significant progress within a short period of time. Also, thank funding agency for their generous support for our different projects. And thank you very much for your attention. I would like to take any questions. Thank you.
Thank you. Let me see if there's any questions in the chat for you. If anybody has questions for Yizhou, please put them in the chat and I will read them. I'll first leave open to your co-presenters to see if they have any questions at all. Sarah or Victoria, do you have any questions?
Yeah, I'm curious if you ever thought of working with an artist.
Sure. I think, in fact, we in our institute, we have Immunology Institute, Brain Institute. We work together with quite a lot of artists, either internally or externally, because we produce a lot of scientific images. We work with artists to share with the broad public, either from the artist perspective or from the scientific perspective. Yeah, we would love to work with artists.
Yeah, so maybe you would be interested in the work of Marta de Menezes. She and her husband, who's an immunologist, actually create amazing, really interesting works. And I'll put it in the chat for you. I think it may be inspiring for you.
Sure, thank you.
Sarah, did you have any comments or questions? No? Okay. I was wondering, these are a really generic question, but what put you on the path to these particular systems that you're working on? How did you get there?
Yeah, that's a long like career path, and because I was particularly interested in development new therapeutic medicine. My undergraduate major was in pharmaceutical sciences. Then I performed my master studies in organic chemistry and PhD back to pharmaceutical sciences and postdoc in chemical engineering and drug delivery. Then I combine all these different knowledge and specialty together, and I found it's a very important field like nanotechnology and RNA therapeutics. That's why I work with our lab members to develop novel technologies and tackle some very challenging diseases. Yeah.
Thank you. I'm also an organic chemist, so I could see the dendrimer in there. Okay, so we have something from an anonymous attendee. Says, your graphic representations in your slideshow are excellent. Do you work with specialized graphic designers with a background in medicine?
Not really. In fact, we have really talented lab members. They, based on our scientific results, and then they produce those different type of images or illustrations to demonstrate our concept or results.
I'll ask one last question, which is just, so what inspired your particular design strategy that you're using? What scientific things inspired you to go to that place?
Yeah, we utilize all kinds of strategies to design new materials or new RNA. One of them it's a really bio-inspired. We try to learn from the nature. For example, a lot of our material components are from cell membrane. For example, phospholipids. We modify a lot of phospholipids. That's a natural component from cell membrane. And the vitamins, those are very important class of the small molecules in our body. We engineer those molecules. So that's a very important strategy. We either are inspired by the nature or we all mimic the nature, and that can help us to either from the delivery perspective or from the therapeutic perspective to improve the pharmaceutical properties of the formulation.
Cool.
Great. Anybody else? Comments or questions? Not, thank you.
Put the links for you in the chat. And anybody in the audience, I don't know if they can access it.
Let me see.
Yeah, I saw the link for the chat. Thank you.
Okay, got it. Okay, thank you.